That's a
copy
probably
your
salad.
because I don't
I'm
have
just kidding, I smell like
recordings.
garlic and onion. I spent all morning cooking chicken, same thing.
Same one?
Well, this one's got curry.
Already?
That's for the invite.
You put it in?
dude.
Hey, it's not done yet. You can come with a snack. You got what's in there?
No.
All right,
Did you
so
get
go
it?
to the left PowerPoint
Anything from the
and then go down to the bottom of lesson system 2. So right down here, the very end after the sample exam,
There's 15
it
people.
says take home. And that just opens up the file, and then you have to back up, and here will be the exam. So because there's just one exam, you're not going to find your name on there. All right? So everybody, what you're going to turn in is the exam itself. You're not turning in a Scantron. So mark your answers on the test. And then you open it up. So
One that looks like a leopard.
you'll see a variety of questions. How do I increase the size of it?
You download it and then you
Does it have like a lynx ears though? Or they like just little hair
There you
thing?
go.
Yeah, if I got a cat, I get one of those.
Okay,
Couple
let me do it a little
smart
bit
too.
more.
Yeah, some of us are drawing today.
All right, so what it is a series of questions. Most of them have 4 choices. Some of them have 2. And so you're going to mark all the appropriate ones. Each answer is worth 1/4 point. All right, so if the voltage gated channel is the correct answer and you mark it, you earn 1/4 point. If the lagging gated channel is the correct answer and you mark it, you get 1/4 point. If the leak channel is not a correct answer and you don't mark it, you get 1/4 point. Okay, so you earn the point for correctly marking it or not marking it, depending on whether it's the right answer or not. Now let's say that leak channel was the correct answer, but you didn't mark it, then that would be minus 1/4 point. Okay, so let's say that only these 2 were right, but you marked all of them. You would have earned 1/2 point. I mean, because you earned 1/4. So you would have earned half of the 1 point that that's worth. Okay? Now that doesn't mean you can go through and mark all the answers. I know what you were thinking. Because some of them, all of them will be wrong. And if you mark all of them, you're going to miss the whole point. Okay? Pun intended. So there could be just one answer. All of them could be right. All of them could be wrong. 2 could be right. 2 could be any combination. All right? Some of the questions only have 2 answers. They're still worth 1/4 point, and that question would be worth 1/2 point. So the way I add them up is that I just, divide your total correct answers by 4, and that's the number of points from those components. Now, some of these questions are relatively easy. You can go ahead and start working on the ones that are on neurophysiology. On potassium channels and action potentials and ligand-gated channels and so on. Of course, there's questions on the autonomic nervous system, which we've just started, and on the eye and the ear. Some of them you can find in your lecture notes or textbooks. Others you'll have to go on the internet. All right, there's questions about black widow toxins and snake venom toxins and how they affect, what channels do they affect, how do they have their, kind of like the tetrodotoxin question that you had on your lab manual. Now, because there's just one exam for all 58 of you, I am not asking you to sign a form that you're doing the work on your own. I kind of expect you guys to talk about this because I know that you learn material when you have discussions. All right? What I prefer, not what I prefer, what I expect not to happen, and this is up to you to ensure that because I'm not going to have any idea. All right? What I expect not to happen is for somebody to spend 28 hours of work on this and someone else say, can I see your exam, and then in 20 minutes copy down all their answers and not do any other work. Okay? So I can't keep that from happening. You're the only one that can do that. And some people have a hard time saying no when someone asks to see your test. So what you can say is there a question you're having a problem with? I'll be happy to talk it over with you. Okay? If you want to spend 30 hours on a test or 20 hours on the test and give away all that work, there's nothing I can do about that. But it's up to you. And so don't ask someone to give them your test so that you can copy their answers, because that's going to make them feel uncomfortable, and they may not feel. You'll both be unhappy about it. They'll be happy saying yes or no. And so I do expect you to work on them together if you want to. You don't have to. But I know that learning occurs in groups. People talk about things and have different ideas, explain things differently. That's fine. Okay? That's a good thing. So it's the not thinking about it and just taking someone's answers after they've done the thinking. That is not helpful to anybody. So most of the questions are in this format. There are 55 points possible from that type of a format. More questions than 55, because some of them are 2 points, just 2 answers. And then there are, there's a diagram. Get down to it here. So this has to do with the autonomic nervous system. We're going to start looking at diagrams like this today and tomorrow. And so it asks you to diagram a cross-section of the spinal cord, draw in, you might have to modify. I've given you the level as the sacral spinal cord. You might have to modify what you see here, but we're going to be doing this in class. Okay? And then whatever's appropriate, include and label from this list over here. So that's worth 10 points. And then there's another 25 short answer. Okay? So this is up there now. Again, the entire test needs to be turned in because that's what I'm grading. So not a Scantron. It'll take me a little while to grade. We're going to Utah for Thanksgiving, so that will be a bit of a drive, and I'll do a lot of it on that. So it's due our last class period before Thanksgiving. I believe it's the 26th by 3 o'clock. Okay? You may turn it in early. That is the day we're having our lab exam, but it's not required to be due that day. If you turn it in early, I will obviously start grading it, but I won't be able, I can tell you your score, but I can't give it back because, of course, everybody else has the same test. So if I mark correct answers on it, that's not going to work very well. It will put you at a disadvantage. So you can turn it in ahead of November 26th. Our lab exam is still on the 19th, but it's almost 3 weeks that you have to work on it. Okay? So kind of set out your time. You don't want to do this the night before. It'll be miserable. This is not a fun test. Okay? You will hate it by the time you're done. I guarantee. But it's a lot easier to do this information this way than try to grasp and understand and everything we've covered so far. Okay? Plus, I can expose you to some of the varieties of effects and diseases and pathologies and stuff that are out there just to make you more familiar with them. So I don't expect you, when you turn the test in, to know intimately everything you've answered, because a lot of that material you will have found other sources for, and you've transferred the information from that source to the test. You still kind of have to think about it, though. And again, as I said earlier, keep a log of where you find the information in case we have a dispute over whether that information is correct or not. Okay? So just on another legal-sized piece of paper, or just copy and paste web pages or articles or sources, textbooks or whatever. And this is the reason why some of them will be pretty obvious, stuff that we've covered in lecture. But if you think you're talking with 2 other people and they have different answers, but you found it in one place, websites disagree. Okay, there's different information that's out there based on different research. People do a research experiment and come up with one answer, and people do another experiment and come up with additional answers or something completely opposite. So it's not always that it's fraudulent. There are, can be different data out there. Okay, any question on that? Any questions? I want a separate test from each of you. This is not a 60-member gang test. It would be easy to grade one test from all of you. But each person is turning their own test, okay? And that's kind of obvious.
Each person, 2 questions, not you. We done. We could be done with this exam today, everyone. You all get together, choose 2 questions,
All right, I keep talking about the autonomic nervous system. Today is our official start of it. I first mentioned it on the first day of lecture when I described the different divisions, similar to what's on the board. We had a little more complicated diagram than this. We had the sensory and the motor division, found both within the central nervous system and the peripheral nervous system. And then under motor, we had voluntary, which is our skeletal muscle, what our next unit is going to be about. And we had involuntary. All right, so our involuntary motor nervous system is what we're studying today, starting today. We talked about the somatic, and we're going to pick that up again when we do skeletal muscle. We'll have our first lecture on it just before Thanksgiving. So in the autonomic nervous system, there are 2 components, sympathetic and parasympathetic. We've spent most of our discussion on parasympathetic, as we talked about the 4 cranial nerves that contain parasympathetic neurons: ocular motor, facial, glossopharyngeal, and vagus. Okay. So we're going to, that was parasympathetic. We're going to look at that today, and then tomorrow we'll look at sympathetic. And then on Tuesday, we'll bring them both back together again and talk about some visceral or organ reflexes. So neurons of the autonomic nervous system, are they motor or are they sensory? Motor. Now, when I was working at the med school, taking classes as well as teaching there, the medical students had a really hard time with this. Understanding that autonomic is purely motor. Do you remember what our modality was? General, visceral, efferent. All right. Now, when you cut a parasympathetic neuron like the vagus, that nerve, not a neuron, but the nerve is going to have a lot of sensory neurons with it. About 90% of the vagus nerve, cranial nerve 10, is formed of actually sensory fibers. But those aren't the autonomic component. All right. We certainly utilize them. On Tuesday, we're going to look at visceral reflexes. We've already talked about visceral reflexes. You guys think of some of the visceral reflexes we've covered? Exactly. So the pupillary reflex, the corneal reflex, there's an accommodation reflex with the changing shape of the lens. So does that have a sensory component to it? Yeah. Pupillary reflex, exactly, had the light, carried the activating the optic nerve. The optic nerve is not autonomic, all right, but it is carrying the sensory changes back to the brainstem, where reflexes formed with the parasympathetic fibers of ocular motor actually changing the muscle contraction. So we have a lot of sensory reflexes. Our defecation reflex, our going to the bathroom, our micturition reflex. When the bladder fills with urine, it stretches. That's sensory. General, visceral, afferent is going to send a signal to the spinal cord that will send motor impulses back via parasympathetic to cause the bladder to contract. So yes, sensory impulses are important, but they are not the motor. They're not the ones activating the gland or the motor. All right, they're involved in the reflex part of it. So in this diagram, and it's a bit small, but it has a lot of information on it. Let me just turn the lights off here. If you recall, and I think this is on page 71, the information where we're starting anyway. The slide itself is different. So we're starting the autonomic nervous system on page 71, yeah. All right, so at the top is the lower motor neuron. And remember the lower motor neuron, see where it says central nervous system here? The cell body of the lower motor neuron is found in the central nervous system. What part of the spinal cord, if we're talking about?
The anterior gray horn?
Exactly, anterior gray horn. That's what makes it gray, and it's motor, so it's in the anterior aspect. It is a single neuron, and we have another neuron activating. So our cortical spinal tract, the upper motor neuron cell body is located in the precentral gyrus. So if I decide I'm going to reach up and try to touch the ceiling, that conscious thought occurs in my prefrontal cortex. It activates my premotor area about what muscles I need to do, and I reach up and do that. Okay, so it's a two-neuron system. The upper motor neuron being found in the precentral gyrus, and the lower motor neuron being found in the anterior gray horn. Isn't it nice to already know something that I say? You actually understand it? We're going to change that in a few minutes. Now, when we look at the autonomic nervous system, the equivalent of the lower motor neuron becomes two neurons. So we have a cell body in the brainstem or a cell body in the spinal cord. It leaves the brainstem, but instead of going all the way to the muscle or the gland that's going to be activated, it forms a synapse with another neuron. So it takes two neurons to do what one lower motor neuron does. And in the autonomic nervous system discussions and lectures, the focus is always on these two neurons, so much so that I think students tend to think that that's both the upper and lower motor neuron equivalent. But we still have a descending pathway from the brain. Does anybody remember what part of the brain controls the autonomic nervous system? Pardon? No, we do have, that's where we have cell bodies of vagus and glossopharyngeal nerve and part of facial. But even that gets controlled from a part of the brain that's up, superior to it. Nope. That's involved too, but not the control sequence.
Cerebellum?
No.
Run that instructions here.
Start naming things.
Okay. Hypothalamus, regulation of the autonomic nervous system was one of the 10 things we were talking about. All right, so if we look back at, so here we have our upper and lower motor neurons for our cortical spinal or cortical bulbar tracts. Upper motor neuron in the precentral gyrus, lower motor neuron in the brainstem or spinal cord. All right. For our autonomic nervous system, the lower motor neuron equivalent has a cell body in the brainstem or spinal cord, leaves the central nervous system, synapses with another neuron, but there are still pathways that regulate that. A lot of visceral reflexes, like I talked about with going to the bathroom, okay, that just requires a sensory input to the spinal cord and a motor output. But from the hypothalamus, we don't have distinct pathways that we name. Well, actually, they are given a very general name, but we don't see them mapped in the spinal cord like we did with spinocerebellar and tectospinal and so on. So, but these are coming from the hypothalamus and function to excite or inhibit those first neurons that are found in the spinal cord. And that's about all I'm going to go into because we're still learning information about that. Last few years is when they actually started identifying, naming them. I'm going to come back to this diagram in just a moment. So these are new slides from this morning at 7:09. But I'm not going to test you on them, okay? I'll put them up online. There's a website that's down here, so this becomes an animated, you can see the arrows moving, essentially. So the paraventricular nucleus is hypothalamus. And here's amygdala and hippocampus and cingulate gyrus are limbic system components. So emotion certainly affects autonomic nervous system. Right? Heart rate can be affected by emotion. Your mouth can go dry being affected by emotion, and so on. Then there's input from other areas of the hypothalamus and also output. So here's vagus, Here's trigeminal nerve and facial nerve, and then we actually have it going down to the spinal cord. This is way more complicated than even I know. I just found this morning. All right. So the major image that I want you to get is the one that's already up here, and the one that I showed you previously. I actually had a question on the paraventricular nucleus on the take-home exam. Just before I published it this morning, I took out 10 questions. So I made it a little bit shorter. And this is one of the questions I took out. But recognize that, again, the hypothalamus is the central area where if we're going to centralize any control over the autonomic nervous system, it's going to be the hypothalamus. And it doesn't do it by magic. It doesn't do it by telepathy. There are structural neurons that connect the hypothalamus to brainstem and spinal cord. So back to this image right here. Okay. We don't pay as much attention to them because of the prevalence of those visceral reflexes, but they are there. Okay. Two divisions of the autonomic nervous system. Today we're focusing on parasympathetic, but we'll mention a little bit about sympathetic. Typically, the parasympathetic, well, we talk about our organs as having dual innervation. That's a key concept to the autonomic nervous system. For instance, the heart. Left alone, in other words, the heart receives innervation. There are nerves, both sympathetic and parasympathetic, that go to the heart. Without any nerves, the heart has certain areas, pacemakers, that are like neurons. They're really muscle cells, but they depolarize on their own. They have open sodium channels, and it's like an hourglass. The sodium ions leak into the cell, and when the level of positive ions in the cell reaches threshold, that cell fires as if it was a neuron and sends ions into nearby muscle cells and depolarizes them, and the muscle contracts, and that spreads throughout the rest of the heart. So the heart doesn't have to be innervated. It will depolarize and spread that current throughout the muscle. If left alone, your heart rate at the rate at which the cells depolarize the fastest is about 100 times a minute. And while that might be an older person's resting heart rate, typically, clinically, the resting heart rate is around 70. So what brings that resting heart rate down to 70? Parasympathetic. Okay? The vagus nerve innervates the heart. However, we do want it to go fast sometimes. So when we are exercising, or when we're scared, or we need to run, then our heart rate is going to go up. And that is done by sympathetic. All right? So it's not the concept that sympathetic makes the heart rate go up and parasympathetic returns it to a resting heart rate. That does happen, but parasympathetic keeps it at the resting heart rate. Well, without any innervation at all, it would be higher. So that's what we mean by dual innervation. Many organs have both sympathetic and parasympathetic, and they have opposite effects. So parasympathetic is sometimes described as the rest and digest. So it slows the heart rate down when we're at rest. It does our normal body activities, and one of those is eating. So it's going to increase enzyme secretion and hydrochloric acid secretion. It's going to make the small intestine undergo peristalsis and move the food through the digestive tract. All right? It's going to activate the bladder, as I said already, or the rectum when you need to go to the bathroom. Those are normal activities. So that's parasympathetic. Sympathetic shuts down those activities so that the skeletal muscle can work for fighting or running away. Now, there are some structures that are done as a help in the reproductive system. Sympathetic and parasympathetic work together instead of against each other. And some structures, like blood vessels and sweat glands, are only sympathetic. Okay? But for the majority of our organs, it's an opposing force done from both of these. Now, in comparison, and again, we're going to do sympathetic tomorrow, most of our parasympathetic fibers that are not involved with the face have very long preganglionic neurons. Now, why do I call them preganglionic? What have we talked about ganglia so far? Okay, cell bodies outside the central nervous system. And we've been looking at sensory neurons, right? So if we drew a sensory pseudopolar neuron, there would be its cell body, and a whole bunch of these pseudo-unipolar neurons formed a sensory ganglion. Notice there are no synapses occurring here. It's just a cell body with the two processes carrying the actual potential through towards the central nervous system. In contrast, our ganglia for the autonomic is a synapse between two neurons. So we're going to have, if this is our central nervous system here, let's say that we're looking at in the brainstem, and we have a facial nerve. We're going to have the cell body of the preganglionic neuron, or the facial nerve, and it's going to leave the the area between the pons and the medulla, and then just outside the eyeball, it's going to synapse with another neuron, which is going to go to the ciliary muscle. Guess what we have? A whole bunch of cell bodies. So that makes this a ganglion. It's an autonomic ganglion. And this has synapses. We have one cell ending and the cell body of another. So that's a key difference in what goes on in these two ganglia. So that's why we use the term preganglionic and postganglionic. Now, I showed you a facial nerve here, which is kind of an exception to our rule. But starting with the vagus and the rest of the parasympathetic neurons that are involved in the ventral and abdominal cavities. For parasympathetic, the pattern is typically a very long preganglionic neuron, because it has to go from the brainstem or the spinal cord all the way to the organ. It synapses with the postganglionic neuron. And this one is microscopically short, because the ganglion is in the wall of the organ. All right? Not in the structures, not in cranial nerves 3, 5, and 7, and 9, but in all the rest of our parasympathetic. So this would occur in the organ wall or in the gland. So a very long preganglionic neuron and a very short postganglionic neuron. It is the cell body of the postganglionic neuron that actually creates the ganglion. That's why it's a collection of cell bodies. So that's the parasympathetic system. Sympathetic can have a variation. Typically, and again, we're going to look at this more tomorrow. Sympathetic will have its cell body in the central nervous system, and it will travel just a short distance out of the central nervous system. It may travel up or down a short distance, but it travels just a short distance out. Then we have two things that can occur here. This is the first one. Then we have a very long postganglionic neuron. Now this is what we find in neurons that go to the body wall, that go to skin and blood vessels. Of muscles. So this ganglion is going to be right next to the spinal cord. It doesn't travel because it has such a short preganglionic neuron. So this is the first example. In the second example, and again, I'm going to leave this till tomorrow to actually describe. We have a neuron that will travel out of the spinal cord, come back to the blood vessels that come off of the aorta, so it comes right back to the midline of the body. And then its neuron is long, but it goes to organs. So this is going to go to thoracic and abdominal organs. It doesn't have to go all the way down to the bottom of your foot. Not quite as long. And the ganglion is going to be in front of the aorta. Okay? These are going to be beside the aorta, the spinal cord. So these particular ganglia are known as perivertebral. And this is in front of the aorta, which is in front of the vertebra. So this is known as a prevertebral. And that's all I'm going to say about it today. We'll get enough of it tomorrow. But just in comparing sympathetic and parasympathetic. Now, if we go back to that previous slide that included the lower motor neuron, the skeletal motor neuron, this one. Notice what they all have in the preganglionic neurons and the skeletal motor neurons, lower motor neurons, all have in common. What is their neurotransmitter? Acetylcholine. So skeletal muscle, let's say that I want to flex my finger. The reason I'm flexing my finger is because the lower motor neuron is releasing acetylcholine that's binding to receptors on my skeletal muscle. Okay, that's acetylcholine. The same molecule of acetylcholine is being released from my preganglionic neuron to my postganglionic neuron. We're going to finish up lecture today by looking at those ionotropic, metabotropic channels we talked about on Tuesday. Interestingly enough, and this is where complications come with medications, acetylcholine is also released from the preganglionic neuron of our sympathetic nervous system. Why does that provide complications? Well, there's a lot of an overlap. If I give somebody, let's say that, remember I said that going to the bathroom is a parasympathetic event, and someone's had surgery, and it's 3 days post-op, and they still can't go to the bathroom. They've been straight cath, meaning putting a catheter in, draining the urine, taking it back out again, rather than filling a balloon so it's there for a while. They've been straight cath 3 or 4 times, and they just don't have the tone of their bladder muscle back yet. So a physician orders a drug that's going to cause the urinary bladder to contract. That drug is going to mimic acetylcholine. Or it may be a drug that, anybody remember the enzyme that breaks down acetylcholine? Acetylcholinesterase. So maybe if we give a drug that inhibits acetylcholine, called a cholinesterase inhibitor, then acetylcholine will stay at the synapse longer. All right? So what are some of the side effects that might occur from having too much acetylcholine?
Increased heart rate.
Increase, well, yeah, because it's sympathetic. We can have parasympathetic effects, we can have sympathetic effects, and we can have skeletal muscle effects. So if we look at, get through this one right here. Where's my picture? Oh, I'll have to get back to it. So if we look at, it's in there somewhere. If we look at the lower motor neuron, too much acetylcholine can cause convulsions. Because acetylcholine causes our muscles to contract. So if I leave acetylcholine and my synapse for too long, it's going to keep my muscle depolarized and it's going to go into spasms. That's not an autonomic effect, but because my lower motor neuron uses acetylcholine, that will have an effect. Parasympathetic. It activates my parasympathetic system. Too much saliva. Diarrhea. My digestive tract is too active. All right. Typically, the sympathetic responses tend to be the heart. So high blood pressure, okay, a lot of sweating are the sympathetic effects. Because sympathetic neurons are activated via acetylcholine as well. So it makes it a little complicated when you think about this neurotransmitter that's involved in all of these pathways. All right. So, looking at where we find parasympathetic fibers, these preganglionic neurons will be located in which structure here? We already know one. Cranial nerves 3 and 5 and 7 and 10 come out of what structure? The brainstem. Okay? So those are going to have preganglionic neurons. And then for some reason, we jump down to the sacral spinal cord. Right? So you can see that in this image here. Here's cranial nerve 3 and 7 and 9 doing structures of the head. And then vagus, because it's our wanderer, provides parasympathetic to thoracic and abdominal organs. All the way over, here's the ascending, you have this diagram in your lecture notes, I believe. Ascending transverse colon, and it stops there. But we have the descending colon and the rectum and the bladder and the uterus and other organs that are in the pelvis. And they get their parasympathetic innervation from the conus medullaris, from the tip of the spinal cord, from the sacral spinal cord region. So parasympathetic is known as the cranial sacral system as well. Sympathetic comes from lumbar, thoracic and lumbar, so it's known as the thoracolumbar. I don't know why we skip all of that, but for the parasympathetic, it's brainstem and sacral spinal cord. Cranial sacral. So let's look at the cranial nerves. Remember, these first three are the exception where the ganglia is not found in the organ. The ganglia is found outside, so that the postganglionic neuron is not microscopic. It's going to be anywhere from 2 or 3, half an inch to 2 or 3 inches long. Whereas vagus and our sacral ones are going to be microscopic. They're in the wall of the organ. Those cells are very short. But these first ones have their ganglia outside the wall. So let's look at the first one. And we see this diagram when we're looking at the trochlear nerve and the abducens nerve, but I kind of ignored the ganglion that's in there. But see the little yellow ball circle? It's actually flat, not a ball, right here. So this is the ciliary ganglion. And this is described on page 73. So do you recognize the brainstem over here? That's what our quiz is on today. All right, so midbrain, that's what our ocular motor nerve, cranial nerve 3 comes from. It's out of the midbrain. So here's the cerebral aqueduct and our superior and inferior pediculi, like the pons there. And so we have the preganglionic neuron for ocular motor nerve. pass through the superior orbital fissure, and then just to the side, to the lateral aspect of the eyeball, it's going to end. It's going to stop, and it's going to synapse with the postganglionic neuron. And that's going to create a ganglion. So this is what makes out the ciliary ganglion. Now, that ciliary ganglion is going to go into the eyeball, and it's going to go to the iris, and it's going to go to the ciliary muscle that affects the shape of the lens. Uniquely, it's going to get there by traveling with sensory branches of the trigeminal nerve. For some reason, all of our parasympathetic fibers in the head get to their destination by hitchhiking with trigeminal nerve. So I'm not going to draw a trigeminal nerve in here right now, but I'll show that to you later. So here comes our postganglionic neuron, and it's going to go to the smooth muscle, the iris, and the constrictor muscle. Okay? I'm not trying to imply that one neuron innervates two here, so we'll give it its own. All right, so this is preganglionic neuron. And this would be the post. The neurotransmitter is going to be acetylcholine here, and binding to the muscle, the iris muscle and the ciliary muscle, is also going to be acetylcholine. Acetylcholine is a neurotransmitter for both, released by both neurons. Because of that, parasympathetic system is also known as the cholinergic system, because of the acetylcholine. So this is from Netter's, but you can kind of see the parasympathetic fibers coming through here, going to the postganglionic, and then postganglionic neurons then going into the fibers. So this is a schematic. This is also from Netter's. Without all those other muscles involved. What nerve is this right here? Trigeminal. Okay. So this black line is our sensory neurons coming from the eye, having their cell body in the sensory ganglia of the trigeminal nerve, and then continuing into the pons. Here is the preganglionic fiber for the oculomotor nerve, traveling out the oculomotor nerve to the ciliary ganglia, and then the ciliary ganglia travels with the sensory fibers of trigeminal to get to the eyeball. Okay, that's going to be the case for all of these ganglia that influence. The sympathetic fibers that dilate your pupil come in, they're coming up from the thoracic level of the spinal cord. So they have a ways to travel. Guess how they get in the skull? They follow the internal carotid artery. So they do get to the eyeball through the superior orbital fissure following the blood vessels. And we're going to see that pattern in the abdominal cavity as well. So that's ciliary ganglia. I want you to associate ciliary ganglia with ocular motor. All right? Then we have cranial nerve 7, the facial nerve. And the facial nerve innervates 3 glands, 2 different types. 2 salivary glands and the lacrimal gland. All right? So these glandular cells also have receptors for acetylcholine. And the acetylcholine, they're not muscle, but the acetylcholine causes them to secrete more tears or more saliva. All right? Anybody else? have two ganglia with the facial nerve. And one of them has two names, so it makes it a little more complicated. Now we have the pons and the medulla. And the facial nerve cell bodies, the nucleus actually crosses both regions, but our motor, sensory is up in the pons, but our motor is actually down in the medulla. Did I start off with red or green? I don't remember. Preganglion is red? Okay. So here's the preganglionic neuron. It's going to go to the lacrimal gland. And it actually goes, there's a gap. I don't know if you remember from the skull. You can see it better in the next image. Let me turn the lights on here. There's a small crack-like space between the maxilla and the pterygoid plates of the sphenoid bone. And that's where this ganglion hangs. So it's coming out of the facial nerve, and then its postganglionic neurons travel with the sensory of trigeminal again to get to the lacrimal gland, with the second branch of the trigeminal nerve actually to get to the lacrimal gland. And so, I'll leave that image up while I put this up here. In this little bit of a crack found between the maxilla and the sphenoid, we have the pterygopalatine from the pterygoid plates, or the sphenopalatine from the sphenoid bone. And then this travels with trigeminal and gets up to the lacrimal gland. So either pterygopalatine or sphenopalatine ganglion. Again, acetylcholine is our neurotransmitter. So if I just dripped acetylcholine on the lacrimal gland, let's say I had a person in surgery or a monkey that was anesthetized, and I opened up the orbit and saw the lacrimal gland and just dripped acetylcholine on it, it would start releasing tears. Okay, as a result of that. Now the other set of parasympathetic fibers from the facial go down to a pair of salivary glands that are found, one's kind of wrapped around your jaw and the other's just under your tongue. So they're called the submandibular and sublingual glands. And these release a thick saliva that helps to bind your food together, not the watery saliva when you smell food or chew gum or something like that. So those also come from facial nerve, but they're going to go down to a ganglion that's on the inside of the jaw, kind of beside the tongue. And here, the postganglionic neuron is going to travel with trigeminal again to get to the submandibular and to get to the sublingual glands. Actually, this first one is sublingual. This is known as the submandibular ganglion. Okay, the facial nerve has two ganglia. So here is the pterygopalatine, and down here is the submandibular. Again, these are excitatory in the sense that it's going to cause the lacrimal gland to release tears and the salivary glands to release saliva. The next cranial nerve, the last one that we deal with in the head, is for the salivary gland, the parotid gland, the one that's really watery. These are the only ganglia for the parasympathetic system that we can actually see. All right, the vagus and the sacral ones are tiny and in the organs themselves. We didn't have to do a microscopic, make a slide, and then we could see them. The last one is the otic ganglia. Remember otic meaning ear, parotid gland. So the parotid salivary gland. All right, can I erase what's up here? Send some of your pictures. Okay. The glossopharyngeal is a little bit lower down on the medulla. Preganglionic neuron is going to come up beside the ear and synapse with the postganglionic neuron. And the postganglionic neuron is not going to go too far. It's going to again travel with sensory for trigeminal and end up in the parotid gland. And that's our otic ganglion. We still have acetylcholine forming the neurotransmitters for both pre and post. So the diagram here, here's the glossopharyngeal, and here's our otic ganglion and the parotid gland. And so it's going to come out here, travel through, and then travel with trigeminal to actually get to the parotid gland. So we have 4 ganglia for 3 cranial nerves. Ocular motor has the ciliary ganglion. Facial nerve has pterygopalatine and submandibular, 2 for that one. And then glossopharyngeal has otic. Right? We don't name any of the other ganglia. So we don't have names for any of the vagus ganglia, and within, we just call them intramural or terminal ganglia. They're within the wall of the organ, but we don't have any specific names for them. Okay? So here's the vagus nerve coming down, and it's going to have branches going to the heart, to the lungs, to all the viscera, ending with the digestive tract on the left side. So the lower portion of the digestive tract and all the pelvic organs primarily get their innervation from the spinal cord, which is what you see down here, okay? The sacral region of the spinal cord. So the vagus nerve, after it innervates skeletal muscles in the pharynx and in the larynx, travels into the thoracic cavity. Here we see it wrapping around the esophagus. So it's no longer just a straight nerve, but it forms branches that actually rotates to anterior to posterior. Passes with the esophagus into the through the diaphragm into the abdominal cavity. And as we'll see tomorrow, it travels with the sympathetic nerves and blood vessels to the organs. So if the aorta is giving off a hepatic artery, vagus nerve will travel with the hepatic artery to the liver, and then within the liver will synapse with its cell bodies. If the abdominal artery is giving off a branch to the small intestine, the superior mesenteric artery, vagus nerves will travel out the superior mesenteric artery. The small intestine is about 20 feet long. So that's how long the vagus nerve has to be to get to the small intestine. Well, not long, 20 feet long, but has to go to all 20 feet of the intestines to get to those muscles and those glands. All right. So that brings us to the pelvic portion, the sacral portion of our parasympathetic area. I already drew vagus for you, remember the first time, but let me just kind of, before we look at these, just so that you have a cohesive component. We'll leave this diagram up here. As far as the brain is concerned, and we'll put vagus. Now, vagus goes to heart wall, to the organ wall. So, I'm going to draw the stomach, because everybody thinks of the heart. So this is the stomach.
Stomach, heart, lungs, everything.
And our preganglionic neuron. It's going to come all the way into the wall of the stomach. And then we're going to have our postganglionic neuron, which is much shorter than I drew it. It's microscopic. So our ganglion is actually within the wall of the stomach, or the heart, or the liver. So this is an intramural ganglion, and we don't name it anything more than that. If we're talking about any organ in the thoracic cavity and any organ proximal to the descending colon. So stomach, pancreas, liver, small intestine, ascending and transverse colon will be vagus. Okay? And then descending colon, sigmoid colon, rectum, uterus, bladder, organs in the pelvic cavity are going to be what we're going to talk about next, the sacral spinal cord area. Yes?
Neurotransmitter also.
Yes, neurotransmitters also acetylcholine. Both here and here. All right, so then now we're going to skip thoracic, cervical, thoracic, and lumbar spinal cord and get to the sacral spinal cord. All right. So I'm just going to illustrate that with a slash. So the rest of the spinal cord, and this is sacral spinal cord. I'm going to draw it this way, and then I'm going to actually draw a cross-section of the spinal cord with you. So now, instead of having the stomach, let's do the bladder. All right? Because that's what's innervated parasympathetically by the parasympathetic nerves of the sacral spinal cord. The bladder is not in the same place as the stomach, but I'm going to draw it in the same place, okay? I do know the bladder belongs somewhere else. And it's not bigger than the stomach either. We also have a gallbladder, so that's why I put urinary bladder. Now, remember, going to the bathroom is a parasympathetic event. Some students find this confusing because people talk about sympathetic is fight or flight. And people can be so scared that they wet their pants. And yet, the bladder emptying is a parasympathetic event. And it took me a couple of years of reading and looking up stuff to finally find nobody had a very good explanation why. Because if we're firing the sympathetic, here's what happens with sympathetic. Sympathetic causes the bladder muscle to relax. Sympathetic causes the smooth muscle that's squeezing the bladder neck so that you don't leak urine to contract. So everything sympathetic is to prevent you from going to the bathroom. We have a voluntary muscle that squeezes as well, which is why you need to go to the bathroom, but you go into a public bathroom and it's people are there and you don't know who they are and you've never used this bathroom before and it's dirty and you're thinking about all sorts of stuff and you sit there for 10 minutes having to go to the bathroom and nothing happens. Okay? That's the voluntary muscle that's not being inhibited so that your urethra actually opens. But sympathetic is the opposite. It's going to relax the bladder and it's going to tighten the smooth muscle that squeezes the neck of the bladder. So how come being scared to death, it should make all of that tighten up even more? The best explanation I got, and Justin Moore, another faculty of ours, disputes me with this, but he can't come up with a better one, is that sympathetic neurons activate blood vessels. Some of them they constrict, some of them they dilate. Typically they constrict the ones that go to our viscera and dilate the ones that go to our skeletal muscle. So you have more oxygen available to the muscles that you need to run or to fight. And the blood not disappears, but the blood is transferred from your digestive tract to those skeletal muscles. All right? So the thought is, and this wasn't my idea, it was trying to find some information about this. The thought is that sympathetic under extreme fear causes the blood supply to the bladder wall to constrict so much that sympathetic neurons lose their oxygen. They can't function. Therefore, they can't keep the bladder wall neck closed. And that's why it opens. Kind of the opposite effect. Okay? Which was as good as any that I've been able to come across with. So it's not so much that the bladder is triggered to contract as the sympathetic nervous system is unable to keep it from the sphincter opening up. All right. But anyway, normally our preganglionic neuron
Sphincter.
is in the sacrospinal cord, and it will come to the bladder wall. Another preganglionic neuron will come to the sphincter. And here's an example of where the same, I want to come into the muscle itself. The same neurotransmitter can have opposite effects. Here's our very short postganglionic neuron, and again, acetylcholine. And we have a ganglion here, and we have a ganglion here. Acetylcholine is a neurotransmitter in both cases. So this is a visceral reflex. All right, your bladder fills up with water. So all three of us yesterday, my daughter and my son, drove down to San Jose to watch my oldest play basketball. They got killed. My daughter played San Jose is a Division I team. We walked into, it's a very fancy stadium. It looks like not quite NBA, but it's a lot fancier than many of the Division II schools, even much fancier than Sac State. And the score was 133-17. I thought. I mean, that's what it said on the screen, was 133 for the San Jose Spartans, and PUC Pioneers were 17. We go and sit down, and their cheerleaders are yelling defense. And really? A score of 117 and you're yelling defense? And then we're about 20 minutes, 15 minutes into the game, and my son says, Mom, that's not a 1 up there. It's tiny little letters, S.J. San Jose State University, all on top of each other. So the score was really 33-17 when we walked in. But back to the story about going to the bathroom. We were in a hurry. We were late leaving. We got there about 7:15, and I don't want to tell you how fast I went to get there, but I was being passed by other cars. All of us needed to go to the bathroom, okay? I stopped at Taco Bell long enough to get food and drink, which made it even worse. It was a 2-hour trip, a little slightly less because I went fast trip to get there. And we all disappeared into the bathrooms once we got there. It took me, not to get, too literal here, but 5 minutes to empty my bladder. So what happens? The bladder stretches. What are our visceral afferents? Stretch and pain. Both of those kind of go along together with a full bladder. All right, so that sensory information travels back to the spinal cord as the bladder is stretching. Our sensory stretch reflex get back to the spinal cord, and an arc connects these two. So when the bladder fills, there's also a connection to the lumbar level that's gone here, but the bladder fills, parasympathetic fibers fire. Here's a cool thing. Well, I know what I think is cool. It's not always what you think is cool, but this is what I'm thinking about when I'm going to the bathroom last night. This is what's happening. In the bladder wall, acetylcholine is causing this smooth muscle to contract. But what happens if this smooth muscle contracts?
You can't pee.
Can't pee because it's being squeezed closed. So the same neurotransmitter is causing this smooth muscle to relax. So same nerves coming from the sacrospinal cord. One causes the bladder wall to contract, the other causes the bladder neck to relax. And you micturate or void or pee or number 1 or whatever it is. Okay? Kind of cool the way the body knows how these are set up. Now, before our break, actually, this is a little complicated. It won't be in a week or so, but it will be right now. So let's take a break now so that you guys can actually grasp it and come back at 10 after. Okay?
Taking our quiz at the very end?
No, not at the very end. This morning I finished lecture at 9:30 and they were ready for their quiz by 10. I was going to do it at 10:15, but they were ready at 10. So I gave like half an hour to 45 minutes to study after I finished lecture. So I lecture with 3 breaks with 2 hours. So I should be done with lecture at 1. With a couple more.
1:30 we'll take.
1:30 or so.
How long is our break?
Okay. 2 minutes.
How many?
Let's try because I read it on the board.
15. All right. You didn't want some nuts?
So if I have mind, cell, body,
You're
these
good.
are light and
So
channels, right?
Get in on some
Very
almonds.
nice, axon,
Get up with your bad self.
So when this
I'm
sodium
gonna
channel
go slit my
opens
wrist.
and the ions come through, it then is going to close, right? And it's in the absolute refractory state. Nothing will open it. But then it becomes in the relative refractory state, a very strong stimulation. So here's my greatest potential. If I have a weak, it will open, it will reach threshold. And when this recovers, it will be no greater potential left in my cell body. This will not open again. Right? Because I won't have it forever, like it was never the same. That's one. That's very right here. Just strong enough to open my motivated channel. But now let's say that I have a very strong stimulation.
Everyone turn in their lab?
So I
Did you make sure we turned it in?
Question.
Something about numbers.
move here.
Did you do number?
Exactly. Some more.
There was one I didn't
But I
make.
didn't have to.
Oh, that's right, okay, sorry.
You want some chocolate?
Nothing happens. Even though it's very strong, I did my absolute.
You want to walk?
So that didn't happen. So they go on by. Then I get into my relative refractory state. I still have so many ions here that when they come, it's a strong enough
Yeah, she
stimulus.
wants to see on that.
That's what you said on the.
Okay?
Joe, did you?
If I have a really strong muscle and that opens and I'm back to my middle.
Time divided by the number of seconds.
So if I draw that out, remember where I only had enough to bring to right now? Even if I have enough, do you like this thing? I'm gonna have
This is my chips here.
So I tried it. It's terrible. Like usually when I take an apple coffee, I would take 1 sip and I'm like,
if I have enough,
I actually drank it the whole time. Oh, Megan, would you stop the camera, please?
It's going to explain this view. So this is the sacrum right here. And within the sacral canal, remember the conus medullaris ends at L1, L2? And then lumbar and sacral spinal nerves form the cauda equina as they come down. So these nerve fibers right here are the ventral rami of sacral spinal nerves that have descended as cauda equina. They are passing through the anterior foramina of the sacrum, coming together, and they're going out the greater sciatic notch right here is the sciatic nerve, going down the back of your leg. So we have that same image on this model right here. All right, so here's it looks solid, but it's not. This is the cauda equina. So these are sacral spinal nerves coming down into the sacrum, and then their ventral rami are passing out the openings of the sacrum onto the wall of the pelvis. And then they'll go out and form this nerve down the back of the leg, the sciatic nerve. All right? So this is where you're looking at right there on the diagram. In fact, that same pattern. So these are the ventral rami here that you see here. All right? So what I want to do now is cut through the cross-section of the spinal cord from which those ventral rami are coming. So rather than the sacral cauda equina, we're going to go up a little bit higher. I'm going to do just half of a spinal cord.
We're just here.
So you don't have this diagram in your lecture notes. Bring some paper tomorrow because we're going to be doing a lot of this diagramming. In your lab manual, these diagrams are in your lab manual, okay, in the lab syllabus. So they are present there. So this is sacral spinal cord, about S2, S3. Notice that my gray matter doesn't have a lateral gray horn. That's because that's found only in the thoracic and lumbar regions. So tomorrow when we do sympathetic nervous system, we will have it there. All right? What cell bodies are forming the anterior gray horn?
Lower motor neuron.
Lower motor neuron. That one that we saw in the chart earlier. So this is a lower motor neuron. It's going to come out the ventral root. And we're going to send it down the leg. So we're going to send it down the ventral ramus into the sciatic nerve and down to a muscle in the leg. So this structure right here is the ventral ramus. Okay? And this would be our lower motor neuron. Now let's draw in our parasympathetic. This is the sacral level of the spinal cord. There are no preganglionic cell bodies in the sacral level. The lowest level of the spinal cord that are sympathetic, the lowest level that we find sympathetic is lumbar. All right, so when you see sacral, there are no preganglionic cell bodies. We will add this sympathetic chain right here tomorrow to our discussion. So we will see postganglionic cell bodies, but we will never see any preganglionic cell bodies at this level. So here is our preganglionic parasympathetic. Is it going to go out the dorsal root or the ventral root? Ventral. Why? It's motor. Exactly right. So we're going to send it out the ventral root. And then it's going to come off the ventral ramus because this is going down into my leg. Parasympathetic fibers are not found in the extremities. They do not go to the blood vessels in the leg. They do not go to sweat glands in the arms. Those are all sympathetic. So it's not going to, we're not going to have any parasympathetic fibers in the sciatic nerve. All right? So notice what happens. Here is the ventral ramus right here. You see this white line here coming off the ventral ramus? And this one here, and this one here. This is where they come from. Here is the preganglionic fiber, and it's leaving the ventral ramus to come into the pelvis. So here is that white nerve that you saw leaving the ventral ramus. This structure right here is this structure right here. Okay, let me turn the lights off just briefly so you guys can see that better. So the spinal cord is up high, but their ventral rami have come down, leaving, so coming out of the spinal cord, we're going to have the preganglionic neuron here. And then it's going to leave the ventral ramus, because the rest of the ventral ramus is going into the lower extremity. It's going to form the sciatic nerve. But this leaves. It's leaving here. It's leaving here. And it comes into this rat's nest of mixed sympathetic and parasympathetic, and then heads to the uterus or the oviduct or the bladder or the rectum. So we have a particular name for these nerves. We don't call it vagus or glossopharyngeal. All of them, as a group, are identified as pelvic splanchnic nerves. That's what's at the top of the screen there. Pelvic splanchnic nerves. Pelvic splanchnic nerves are formed of preganglionic parasympathetic. I'm not quite done with the diagram here. So we were talking about the bladder. So let me pull the bladder back in place here. It's not going to be nearly as large, but it will be out of proportion. So this is passing into that rat's nest, and there's sympathetic and parasympathetic, a whole mishmash. But this is leading directly into the bladder wall. Again, my proportion is off. So that entire preganglionic neuron bundle is called the pelvic splanchnic nerve. And then within the wall of the bladder or the uterus or whatever organ I'm talking about, we have our postganglionic neuron. And here's the ganglion that I drew earlier. So we have named nerves. Remember I said parasympathetic was craniosacral? We have named nerves for the cranial portion. Glossopharyngeal, oculomotor, vagus. Here, they're all called pelvic splanchnic. Now tomorrow we're going to look at the sympathetic fibers that are coming in here. They're coming off of our sympathetic chain. They're also called splanchnics. They're called sacral splanchnics. And for a while it was like, well, this is coming out of the sacral spinal cord. Why don't we call them sacral splanchnics? And I could never remember which was which until I hit upon the perfect solution, which I will share with you, free of charge. pelvic splanchnic, PS, parasympathetic. Okay? Sacral splanchnic that we'll do tomorrow is SS, sympathetic. You'll never forget that. Okay, at least I hope not.
Forgot it already.
I'll teach it to you again next week. And the next week. Alright, so remember parasympathetic is cranial sacral, and this is the cranial portion of parasympathetic. This is how we get acetylcholine as a neurotransmitter to the lower digestive organs, sigmoid colon, rectum, bladder, uterus, oviduct, seminal vesicles in the guys. This is part of the diagram, not the complete one, but this is part of the information you need for the diagram on your take-home test. And I ask you to draw the sacral spinal cord.
So the red one will be upper.
The red one is the preganglionic parasympathetic. It's not the upper. Remember, in the autonomic nervous system, it takes both of these to really be the lower. But it is the one in the central nervous system. So next semester, if you take 431, you will already know this. Well, at least you'll know where to go look for it again. And it won't take you as long to relearn it because this is going to be involved with the micturition reflex, for defecation reflex, for how we do erection and ejaculation when we talk about the reproductive system. So you'll understand the basis of where this is coming from. And we don't really go into any more detail than what we're going to cover this week. All right. So now let's look a little bit at this is the anatomy. Let's finish up by looking at the physiology of the, you don't have this as a list in your lecture notes, but it is in the PowerPoints, and we're going to go over this on Tuesday when we tie everything together with the autonomic nervous system. But this is a summary of what I said earlier about the parasympathetic being the rest and digest, the maintaining of homeostasis. So breathing, digesting food, going to the bathroom, lowering our heart rate, all of those structures I talked about, functions I talked about earlier at the beginning of class. So how do we do this? May I erase what's almost after you? Anyone else that needs to take a picture of what's on the board? And we're going to go back to what I talked about on Tuesday. Nothing that I'm going to talk about for the next 5 minutes is really new. A couple of new words, but no new structures. Okay? We're going to talk about the receptors for acetylcholine. And we have two different types of receptors for acetylcholine. Anybody remember what they are? What were the two different types of ligand-gated receptors that we mentioned? Ionotropic?
Metabotropic.
Metabotropic. Okay, so we have those types of receptors for acetylcholine. One is in the postganglionic neuron, and one is in what we call the effector, the muscle or the gland. All right, so one is released by the preganglionic neuron, and one is released by the postganglionic neuron. Because remember, acetylcholine was used for both. So the preganglionic neuron I'm going to draw this a little bit larger over here, is going to form a synapse with the postganglionic neuron. And that is going to release neurotransmitters. Well, let me bring this around so you guys in the back can see it. So at this level here, we're going to have, again, what is known as the effector. So this would be a muscle cell or a glandular cell. And it's also going to have a receptor. So preganglionic neuron. Parasympathetic. It is also our presynaptic neuron, right? Because it's the one releasing the neurotransmitter. And this is our postganglionic neuron. And it is also forming our postsynaptic membrane. So we have a synapse here in our ganglion, and we have a synapse here with our muscle or gland. Does that make sense? So preganglionic and postganglionic, what I was showing you in that long, I'll go back to this original diagram. Okay, so our first synapse here is this one, between the pre and the post. And our second one is in the organ, this urinary bladder muscle or the heart muscle or the ciliary muscle in the eyeball. Okay, everybody kind of situated with me? So that this first cell, and I'm going to draw it larger now. Here's the synaptic bouton. Of the preganglionic neuron. Releasing acetylcholine. Here would be the cell body, or the dendrite, of my postganglionic neuron. And it's going to have a receptor for acetylcholine. This receptor is an ionotropic receptor. What does that mean? The receptor actually has the channel for the ions. So it's ligand gated, but when the acetylcholine binds to it, it will open. So the postganglionic neuron contains the ionotropic receptor. When it opens, for acetylcholine, that is, and it binds acetylcholine. So we're just talking about acetylcholine, not any ionotropic receptor, but we're talking about acetylcholine. When it opens, it is a channel for sodium ions. So when it opens, sodium ions are going to diffuse into the cell. Is that going to create an EPSP or an IPSP? An EPSP. So this is always excitatory. Now, these particular ionotropic channels that bind acetylcholine are also known as nicotinic receptors, because experimenters, medical researchers, looking for why things worked in the brain the way they did, used a variety of different chemicals to throw it on the brain and see what happened. And they found that nicotine would bind to these acetylcholine receptors, but nicotine would not bind to the acetylcholine receptors on the glands or on the smooth muscle. So these are preferentially, so they are called nicotinic. So acetylcholine ionotropic receptors are classified as nicotinic. If we add nicotine, we get the same effect as if we add acetylcholine. All right. We find these types of receptors on 3 different locations. What I showed you in that previous slide. So they are on all of our postganglionic neurons. They are on parasympathetic postganglionic neurons right here. And as we'll see tomorrow, they are also on sympathetic postganglionic neurons. They are also on skeletal muscle cells. So if I have too much acetylcholine, I get parasympathetic effects, I get sympathetic effects, and I get skeletal muscle effects. Too much acetylcholine on the skeletal muscle can give me convulsions. My muscles are contracting from all that acetylcholine stimulating the muscle cell to open. Parasympathetic effects, increasing my digestive activity, vomiting, and diarrhea, and excess saliva. Those are the parasympathetic effects. Because I'm also activating my postganglionic sympathetic neurons, that tends to be the primary response with the cardiovascular system. So I see high blood pressure and a rapid heart rate as well as sweating. So those are all because of the sympathetic, because acetylcholine is a common neurotransmitter for all 3. So it can be very dangerous to give someone too much of a cholinergic drug. So here is our ionotropic channel. We looked at this on Tuesday. Acetylcholine binds, the channel opens, and positive ions rush inside, making the inside of the cell more positive, as you correctly identify, creating EPSPs. So again, these are always excitatory. Right? We never turn off the postganglionic neurons by firing the preganglionic. So then the action potential created by the opening of these excitatory channels travels down the nerve axon and releases acetylcholine at our synapse or neuromuscular junction. And these channels now are metabotropic. So the channel binding acetylcholine on the smooth muscle or on the cardiac muscle or on the gland are the metabotropic ones. Remember, those are the ones with G proteins. Are those always excitatory or always inhibitory? Third question. Probably if you look at most, it's excitatory in the brain. If you look at most of our receptors, they're probably mostly excitatory. We have more receptors in our digestive system, and it's excitatory for all of those. It's inhibitory in the heart. Okay? It's inhibitory at that sphincter of the bladder, but excitatory to the bladder wall. So it depends. So these are the metabotropic. Nicotine won't bind to them. Instead, a component of a mushroom was found that would. And so these are called muscarinic. Those are the 2 new words, nicotinic and muscarinic receptors. We have a variety. They're called M1, M2, M3, M4, M5. And they have different effects. Okay? M1 happens to be an inhibitory pathway. We'll look at that in just a moment. So they're G proteins. We find them on skeletal muscle, on our smooth muscle, and we find them on our glands, and we find them on some sympathetic, like sweat glands. It's kind of weird that we actually have them at those locations. Notice the effect of too much binding of metabotropic is, except for the saliva, except for the sweat, which isn't listed here, is all parasympathetic. Because this is where we have our sympathetic cells are going to be releasing norepinephrine. So if we drip acetylcholine on our lacrimal gland, we're going to get a parasympathetic effect. All right? If we drip acetylcholine on our sympathetic postganglionic neuron, we'll get a sympathetic effect. So it depends on where that acetylcholine is. If it is on the effector, it's going to be a parasympathetic effect. So here's M1 receptor. This is just no channel at all. That's why it's a metabotropic. Here's our G protein. Okay. The alpha unit with an M1 receptor, the alpha unit is excited. It binds to phospholipase C. Do you remember that pathway that we looked at? And causes PIP2 to split. The most important part here is the diacylglycerol. It activates an enzyme that causes potassium channels to open. If that was what was happening here, would we have an IPSP or an EPSP? An IPSP. Because we're losing positive ions, the inside of the cell becomes more negative and less likely to fire. If this was in the heart muscle, the heart rate would slow down. If this was in the constrictor muscle around the urinary bladder wall, it's going to relax and open. Okay. So this is when we see an inhibitory effect of acetylcholine. Sometimes we see an excitatory effect in the pancreas, in the small intestines. We see an excitatory effect here, where it is the gamma subunits cause the sodium channels to open, and that increasing positive charges results in the potassium channels closing, and the net effect is the inside of the cell becomes more positive. We stop losing potassium, and we gain a whole lot more sodium. The inside of the cell is positive. Sometimes even calcium comes in. If that's the case, it makes smooth muscle cells contract. So again, whether acetylcholine has an excitatory or inhibitory effect depends on the receptor and the downstream events, not on the acetylcholine. That's the significant thing about the metabotropic. It's what the receptor causes to happen, not the acetylcholine itself. That determines what's going to happen at the muscle cell, whether it's going to be more active or less active. Alrighty, let's stop there. Tomorrow, there's some drugs that I have listed. This is a mnemonic, if you will, for the excessive metabotropic effect of parasympathetic. And then we'll kind of tie this in together on Tuesday when we look at how we deal with these things. You have about a half an hour or so. I'll check with you about 5 after, 10 after 1. See if you're ready. If you want to go longer, 10, 15, for our quiz. Did all of you get my announcement on D2L?
About the quiz?
The quiz covers the brainstem model. So that little popsicle looking thing. Remember, the brainstem model includes the parts of the brainstem. It also includes the diencephalon. So we have the thalamus and hypothalamus and the pituitary and amyloid bodies and pineal gland and the insulas all on that model. And there is one question on the cranial nerve. So all the cranial nerves on there, but the olfactory. So actually, the quiz is on everything but the cerebral lobes. That's all on that particular model. So there's one question on CSF spaces that we see on that model, one question on cranial nerves, and the rest are on the bumps and the things that you see on the model. Okay? So I'll check with you in about half an hour. See if you're ready.
Can you go back to the muscarinic receptor?
That.
Yeah.
Okay.
Stop. Stop. I'm fixing my answers to my mom's pencil. Like, even though I was pushing hard, it was still really light.
Probably because it was that hard pencil lead and then it's solid pencil lead.
It was a light marking pencil.
Еще пока не